Alright, so let's talk about the plasma, the fluid portion. So this is the ground substance of blood. Remember, blood is a special type of fluid connective tissue. And then the ground substance, which is the fluid portion of a connective tissue, is plasma in blood. So it's mostly water. And then you also have electrolytes as well. So things like sodium, potassium chloride, calcium ions. And then you have many types of nutrients, and if you don't know what the basic types of macronutrients are, go back to chapter 3, I believe, or was it chapter 2? Whichever one had like the macros. I think it was chapter 2, near the end. And also organic wastes and also proteins. Now the thing is that water and electrolyte balance, why is that important? Well, it all deals with osmosis, and if you don't know what osmosis is, review I think it's maybe in the cell biology chapter when we talk about transport across the plasma membrane. Osmosis comes back with a vengeance this semester, especially when we get to the kidneys and the urinary system. And actually, even in like part 2, or for exam 2, when we talk about hemodynamics, you also have osmosis as well. So you need to if you don't have a good solid understanding of osmosis, go back to that. But why does that matter? Because again, thing is that with your cells and the surrounding extracellular fluid surrounding them, you always have water moving into cells and out of the cells. And if it's kind of balanced where you have roughly equal amounts of water going into a cell as you have water flowing out of a cell, this is what you call an isotonic solution. So the cells stay the same volume because it's receiving as much water as the water is leaving the cell. But if you have a hypertonic solution, so this is like a solution that's very salty or has a lot of glucose or has a lot of other solutes or even proteins in there, what it does is kind of like sucks out the water. So it's like when you pickle something, you have like a high concentration of salt, so that helps to draw out the water from whatever you're pickling. Well, that's what happens in a hypertonic solution. And a hypotonic solution, so a hypotonic solution, relative to the inside of a cell, this has less solute than the inside of a cell. So what happens is that instead of drawing out water, you have water rushing into the cell. So now the cells have a higher solute concentration than the surrounding solution. So eventually all this water going in is going to cause these cells to swell up, and we see one cell here starting to burst. Alright, so then what are our biological macromolecules, especially when we talk about nutrients, which we will, I think, for exam 3. And one way to know three of the four major types are the ones on the back of your nutrition label. Again, we have things like fats and lipids, we have our carbohydrates, our sugars, and we have our proteins. So these are things that are also transported in a smaller molecular form in our bloodstream to help nourish our cells and tissues. Also organic waste, and there are many types of organic waste. Do I expect you to know every single type of organic waste? No, there's too many. But a big category of them are nitrogenous wastes. And why are they nitrogenous wastes? Well, examples of nitrogenous waste, I'm just showing you the chemical structures. Now, you don't need to know these chemical structures because, again, not a lot of you have, I'm pretty sure only a small portion of you have had organic chemistry, but these are the structures of these various nitrogenous wastes. These are compounds your body is trying to get rid of. So you have things like urea, uric acid, creatinine, bilirubin, ammonia. But what do they all have in common? They all have this nitrogen. So this is why. And the funny thing is, like, if you go on to organic chemistry or work with a lot of nitrogenous compounds, they tend to smell kind of fishy and pungent, and that's due to and this is why like urine also has its smell, because of all these compounds, or part of the bouquet of chemicals you get from the smell of urine is from all the nitrogen you have. But again, you're trying to get rid of that. So many different types of proteins in your plasma. Albumins make up the majority of the blood proteins in your plasma. So remember, like, blood is a connective tissue, and then instead of having solid extracellular fibers, you have plasma proteins, and albumins are the majority. And then you also have things called, another category called globulins, and then you have fibrinogen and other proteins that make up that. And what's one example of this less than 1% that's still important? Well, many of your peptide hormones, they're still proteins. They don't make up the majority of proteins in your plasma, but they're still there and they still have those effects. So actually, not just hormones, things like clotting factors, cytokines, chemokines, and there's like hundreds of cytokines and chemokines. So you don't need to know this laundry list exactly, but you should know general categories. So what are albumins and why are they important? And why do you make so many of them? So things that they maintain something called oncotic pressure in blood. And when we talk about blood pressure, you do need to know about these albumins. I think will be possibly on exam 1 and exam 2. So oncotic pressure, if you don't know what osmosis is, please review the previous lectures. But oncotic pressure is referring to a specific type of osmotic pressure due to proteins. So osmosis is a force that moves water, but now proteins are driving the movement of water. So if you've taken the lab or have done this in high school, like I think even for AP Bio I did this in high school. But what you saw is like two solutions. They have different concentrations of salts or sugars, and you see which way the water moves across this dialysis bag, which is acting like a cell membrane. So what we have with serum albumins is that you have two solutions, and here we have, or in this example, we have the blood, and we have interstitial fluid, and then we have a membrane between, or we have a barrier between these two. So these are the membranes of the cells that are between the blood and other fluids in your tissues. So if you have a lot of these proteins that contribute to something called oncotic pressure in your blood, and not so many of these proteins in your interstitial fluid, which way will the water go? Well, to think about it this way is to think about osmosis. If we replace these proteins over here with like sodium chloride or sugar molecules, say there are more sugar molecules in your blood and less sugar molecules in your interstitial fluid, which way will the water go? And I already see a response, and it looks like somebody says it'll go to the blood, and yes, you are correct. These proteins, they have all these charges, the positive and negative charges, that are very attractive to water. So serum albumins are a very important way or important mechanism of how your blood is able to hold on to water. These serum albumins are very attractive to water, so it allows your blood to retain water compared to something like the interstitial fluid, which doesn't have these albumins, or as many of these albumins. So albumins are produced by that very important and I think undersung organ in your book called the liver. So the liver produces a lot of these albumins, but what happens? Again, remember these albumins are important for holding on to water and drawing water into the blood. But what happens if something happens to your liver, or if your liver gets diseased, or your liver isn't functioning as well as it used to? Well, what happens if the albumin concentration drops off like this? Now, let's ask the chat. We had a lot of albumins in our blood, but all of a sudden something went wrong with our liver, and then now we have a lack of albumins. Which way will the water go? Into the blood or into our tissues? Yeah, so it looks like everyone's saying into the tissues, and you're correct. So you have a drop in oncotic pressure, and then the water, instead of going to the blood, now that we have more proteins in the tissues and less protein in the blood is going to lose its ability to retain and hold on to the water. So now this water is going to leave the blood and go to the tissues. And if this happens in the abdomen, this is what we call ascites. And this is what we see here. This person, notice that their limbs are pretty skinny, but this is not just having a big beer belly. This is all this fluid accumulating in the abdomen due to liver dysfunction. So when someone has liver failure, this is a common thing you can see in ascites, is something you commonly see in liver failure. And also edema. So edema is fluid accumulation in connective tissue and peripheral tissues. So here's a specific type of edema called pitting edema. So what we see here is that this is someone pressing into the tissue, the foot right here, and these indentations are staying there because this person has so much fluid that it's just staying there. It's not being reabsorbed by the veins in their circulatory system. So how do you treat this? I think this is something we're going to cover in the next exam when we talk about the or your arteries and veins, or actually even in like exam 3 when we talk about the urinary system and fluid balance. So notice that osmosis is involved in all of this, right? So there are various things you can do to affect your fluid balance by changing the osmotic and oncotic pressure of your blood and your fluids in your body. So you can get rid of fluids, but you can also like, are you going to introduce fluids back into the body and surrounding tissues, or are you going to draw fluids out of the body? So yeah, that's why things like hypotonic, hypertonic, and isotonic become very important, especially when you're using IV.